Almost everything your body does is controlled by DNA sitting in the nucleus of your cells. But there's a second, much smaller genome hiding inside you, one that came from an entirely different organism billions of years ago. In 2015, scientists found a hidden gene inside it that nobody knew was there. It codes for a tiny 16 amino acid peptide called MOTS-c. It doesn't come from your main genome at all. It comes from your mitochondria. And it might be one of the most important signals your body sends out that almost nobody's heard of. Today, what MOTS-c actually does, what the real data shows, and why 2026 might finally answer whether any of it holds up in humans. MOTS-c stands for Mitochondrial Open Reading Frame of the 12S rRNA-C. The important part, a short peptide, 16 amino acids, encoded inside your mitochondrial genome, not your main nuclear one. First characterized by Dr. Chang-Han Lee and colleagues in 2015. Before that, mitochondria were mostly seen as cellular power plants. MOTS-c was some of the first solid evidence they also send their own hormone-like signals to the rest of the body. Did you know? Your mitochondria are widely believed to have started as free-living bacteria roughly 2 billion years ago, before being absorbed into a larger ancestral cell and becoming permanent. That's why they still carry separate DNA today. Part of your genetic code has never fully become you. It's still running its own genome in every one of your cells. MOTS-c isn't the only signal your mitochondria send out. It belongs to a broader family called mitochondrial-derived peptides, or MDPs, including a peptide called humanin, and a group called the SHLPs, short for small humanin-like peptides. This whole category of biology is young. Researchers are still in the early mapping stage of what each signal actually does. Here's the core mechanism. Your cells have a master energy sensing switch called AMPK that normally activates when energy runs low. MOTS-c appears to activate that same switch through a different route by influencing the folate cycle, which raises a molecule called AICAR, and AICAR turns AMPK on directly. The key detail, this happens independent of whether your cell is actually low on energy. Think of AMPK like a backup generator switch. It usually only kicks on when the power goes out. MOTS-c seems to flip that switch anyway, rehearsing the emergency response before there's a real emergency. Did you know, under cellular stress, MOTS-c doesn't just stay near the mitochondria. Research shows it can travel into the cell's nucleus and interact directly with gene expression, a signal starting in your mitochondrial genome, reaching into your main genome. That matters because it reframes what this peptide might be doing. If your mitochondria are a power plant, MOTS-c under stress is like a messenger running straight to City Hall to change policy, not just patching a power line, but potentially rewriting how the whole cell responds to stress, antioxidant defenses included. Here's where it gets interesting for exercise physiology. In animal studies, MOTS-c behaves like what researchers call a mitokine, a mitochondria-derived signal released during physical activity. As muscles demand more energy, MOTS-c has been observed rising alongside that demand, tracking with increased glucose uptake into muscle tissue. To be precise, this timeline is mapped in animal models. Human timing isn't fully confirmed. But the pattern suggests MOTS-c might help coordinate your body's real-time metabolic response to physical stress.
Did you know, some studies observe circulating MOTS-c levels declining with age. Around the same period, metabolic disease risk climbs. That correlation drives a lot of the attention in aging research, though correlation alone doesn't prove the decline causes those changes.
So what does the research actually show? In diet-induced obese mouse models, exogenous MOTS-c improved fasting glucose and glucose tolerance. In a mouse model of heart failure from pressure overload, it reduced inflammation, boosted antioxidant capacity, and preserved cardiac function, tied to that same AMPK activation. Separate research shows MOTS-c restoring mitochondrial respiration, specifically in diabetic heart tissue. And 2026 studies found protective effects elsewhere, preserving cell survival under oxidative stress in reproductive tissue and improving tissue survival in ischemic flap models by reducing specific forms of cell death. Important flag here. Everything covered so far is animal or cell culture data. Genuinely promising, mechanistically consistent, published in real peer-reviewed journals. But mouse and human biology don't always translate directly. That gap is exactly why the next part of the story matters. Did you know, at just 16 amino acids, MOTS-c is remarkably small compared to most signaling peptides in the body? Its size is part of why researchers think it may act quickly and diffuse easily between tissues. Still an active area of study, not a settled fact.
This is the real headline of 2026 for MOTS-c. A Phase 2a human trial is now underway in people with prediabetes, testing skeletal muscle glucose metabolism and AMPK-related signaling directly. This doesn't prove MOTS-c works in humans. That's not what a Phase 2a trial does. What it gives researchers is a real framework, measurable outcomes in actual human physiology, instead of another round of mouse data. That's the difference between a promising mechanism and an actual answer.
Did you know? Phase 2a sits fairly early on the drug development ladder. It typically follows basic human safety testing and looks for an early efficacy signal in a small group, not final proof. Several more stages would be needed before any approval conversation could even begin.
Worth addressing directly because it comes up constantly in online peptide communities. MOTS-c is not a research-backed alternative to drugs like Ozempic or Zepbound. Those target hormone receptors, backed by completed large-scale human trials. MOTS-c works through a completely different mechanism, cellular energy sensing, not appetite suppression. And today, there is no completed human trial showing meaningful weight loss from it at all. The excitement is about mechanism and animal data. The human evidence is still being built, starting right now. Did you know, in biohacking communities, MOTS-c is frequently combined with other unproven compounds in self-directed longevity stacks. Stacking multiple unregulated substances multiplies the unknowns, not the benefits. There's no combined human safety data for any of these pairings. So where does that leave access? MOTS-c is not FDA approved for any human use, with no approved medical indication anywhere. It's sold online, explicitly labeled for research use only, not for human consumption. A label that exists precisely because these products haven't been evaluated for human safety, purity, or dosing outside a controlled trial. People sourcing and self-administering it outside that framework take on real, unquantified risk. Unverified purity, no established human dosing, no medical supervision, and genuinely unknown long-term effects. That's a meaningfully different risk category than a supplement, an unregulated, self-injected research compound. Did you know a lot of the excitement around peptides like MOTS-c comes from correlational findings? Levels are lower in older or metabolically unhealthy populations. Observing a correlation is a far weaker claim than proving restoring those levels actually reverses the problem. That distinction gets lost constantly online. MOTS-c sits in a genuinely fascinating spot in metabolic science, a real peer-reviewed discovery with a mechanism that keeps showing up in serious journals, now finally reaching a real human trial. It also sits in a spot where public excitement has outpaced the evidence, a pattern worth recognizing whenever a compound gets discussed heavily online before clearing even one completed human efficacy trial. The right way to engage at this stage? Watch the research, not self-experiment with an unregulated compound sold explicitly as not for human use. So here's where this leaves us. MOTS-c is a genuinely novel discovery, a peptide encoded by the part of your DNA that isn't originally even human, activating a stress response pathway that reaches into your main genome with real, published, peer-reviewed animal data behind it.
It's also, right now, an unapproved research compound with no completed human efficacy trial, sold explicitly outside any medical framework. Both things are true at once. If you remember one thing, a fascinating mechanism and a proven human treatment are not the same milestone, and MOTS-c has only just crossed the starting line of finding out which one it actually is. Thanks for watching. Subscribe, because when that Phase 2a data lands, we'll cover it here first.